Okay, we're going to attempt to go through this quiz 2. Let's see if I can get this working. There we go. I can't do full screen and this at the same time, so I will just leave it like this. Okay, so some of these quiz questions you've done really well on, so I'm not going to spend too much time on. Chapter 1, a haploid cell has only one complete set of chromosomes. That's kind of the definition of a haploid cell, is that it's a gamete, provides one copy of each chromosome, and then when fertilization occurs, that's when you become a diploid and have homologous chromosomes for each. But a haploid cell has only one complete set of chromosomes. Number 2, a cell in the G0 stage has withdrawn from the cell cycle. So the GO stage is kind of specialized stage where the cell is not growing, and its purpose is not to eventually it's kind of exited the cell cycle in order to kind of stay how it is. S phase is when it's replicating its DNA synthesis. It's not in the process of dividing. Actually, a cell goes into G1 actually after it has just completed cell division, and then GO is a specialized version. In what stage does a tetrad form between two homologous chromosomes? Prophase 1. We were told that the genome size of the C. elegans nematode is 100 megabases. At the beginning of prophase and mitosis, how many megabases of DNA are found within the cell? So this is a straightforward application of what you've done in the activity. And so just to remind you, the genome size is what is present in a haploid cell. So this would be a sperm, and the sperm has approximately 100 megabases. In order to create a fertilized cell, the sperm has to find the oocyte, and so the oocyte also has 100 megabases because it has its own set of copies of each chromosome. And so when you combine these together, then you end up with a fertilized cell that has 200 megabases of DNA. However, at the beginning of prophase and mitosis, we can't just count on the fact that these two things have come together, because now we've also gone through S phase, and so the DNA has actually duplicated. And so because the DNA has actually duplicated, we further have to multiply this number by 2. And so the answer is 400. So there's 400 megabases of DNA within the cell in prophase. Again, because DNA has already been synthesized, and now we have 2 copies of everything. Question 5, normal meiosis takes place in a diploid cell of genotype ABB. So this is how we designate between the 2 chromosomes. And we assume no recombination is possible. So which of the following genotypes could be present in a daughter cell, assuming no errors occurred? So, if we started out, they're in the beginning of meiosis 1, prophase 1 would occur. So kind of how we're writing this out is, how can I change color? Can I change color? Well, the tetrad has formed between them, and so this would be A. This guy would be A right here, and then this guy would be A right here. So in this case, the chromosome is named big A, and this homologous chromosome is named little a. And then we would have something similar going on with the other chromosomes with a big B, little b. So after meiosis 1, the purpose of meiosis 1 is to separate homologous chromosomes from each other. And so you can exclude all of these answers right here that have 2 versions because you've separated the homologous chromosomes out. And so the answer to this one is either this. So because of independent segregation, the law of independent assortment, A and B are separate from each other, and so you could have big A, big B, or little a, little b. There's also, additional answers, so big A, little b, and little a, big B would also be possible. Number 6, normal mitosis occurs in a diploid cell of genotype big A, big B, little b. Now what happens in mitosis? So this question is easiest to answer just if you remember that. Mitosis is a conservative process, so the goal of mitosis is to create exact copies of DNA, and so the result will be the same as the starting genotype. And so the answer is big A, big B, little b. 7, in mitosis, sister chromosomes are separated during anaphase, and then in meiosis, sisters separate in anaphase 2. So the correct answer is D. So anaphase 1, the important thing to remember with that's when the homologous chromosomes separate, and then anaphase 2 is when the sister chromatids separate. 8, during oogenesis in animals. So we covered this part in discussing defects in meiosis and how chromosome non-disjunction can actually lead to things like miscarriages and diseases like Down syndrome. And so we covered the process of oogenesis, and one of the things we discussed is that it starts in the uterus, so in the fetus. So the fetus starts to produce all of the cells that will eventually become oocytes, but that actually pauses, and so it doesn't restart. Oogenesis doesn't restart, for approximately 13 years. And then when an egg is released in a female, then you continue oogenesis. So the correct answer is there can be very large gaps, years between one stage and the next. B was commonly chosen by students as well. Only one cell gets chromosomes, the rest are polar bodies, and so that's close. So one of the important things to remember about oogenesis is that polar bodies are created, and these polar bodies don't actually create oocytes. But the reason that this answer is wrong is because those polar bodies also get chromosomes. So the chromosomes are segregated. You still have to have the total amount of DNA to create a haploid cell. So the polar bodies actually get chromosomes. And so that's why it's incorrect, because it's not true that only one cell gets chromosomes. Only one cell actually develops into an oocyte. This picture depicts a cell that started out as 2n equals 4. So what stage of mitosis or meiosis is it in? And so the answer to this is metaphase. So you should have realized it's not prophase because everything's at the midplane. You see all of the microtubules have formed. So prophase is not a good answer. And the reason metaphase 1 is not an answer is because you should see a tetrad. So we don't see a tetrad here. And the reason metaphase 2 can't be correct is because we know that it's a 2n equals 4. And so you can see that the homologous chromosomes are two copies of homologous chromosomes are still present within the cell. And so we know that is not the case in metaphase 2 because we've already separated homologous chromosomes out to different cells. So you wouldn't see one of the 2 copies of this. So if the cell looked like this, where you had one blue copy of the big chromosome and one orange copy of the orange chromosome, you knew it was 2n equals 4, then you would know the answer is metaphase 2. But because that's not what's shown, then the answer is B. So a decent number of you chose E. So I just thought I'd highlight that. 10, if the cell was isolated from a diploid organism, it is probably in what? So you should immediately get rid of prophase because you can see recombination has occurred. Big orange, there's orange parts of that. And then the other reason that you can exclude prophase 1 is because you would expect to see a tetrad. So we don't see a tetrad here. And so in fact, we see homologous chromosomes have already separated from each other. So there's only one copy of each chromosome. And so C is the best answer. You don't see chromosomes in S phase or G2. 11 is a little bit tricky. So this is a scanning electron micrograph. So this is depicting cells in what stage. So this is kind of similar to the way you do a karyotype. So typically you actually lyse and release the cells from the nucleus. So sometimes the best approach to these questions is just to exclude answers. So we know it's not S phase and we know it's not G2 phase because the chromosomes aren't condensed during those phases. So those are parts of interphase. And so now we have to choose between prophase 1, metaphase 1, and metaphase. And so the way to answer this question is actually you should be able to exclude prophase 1 and metaphase 1, and that's because you don't see a tetrad. So a tetrad, again, remember, is held together by proteins, and so even if you lyse the cells, you would still expect to see the homologous chromosomes paired together. And so the best answer for this one is E. And then 12, this is a matching question. So you had the following phases: prophase 1, metaphase 2, etc. And so you're trying to figure out the answers, the best answers for each of these depictions. And so you have centromeres. So the homologous chromosomes are things to pay attention to, whether or not the sister chromatids had separated from each other or not. So looking at F and G, the way I would look at this answer is to say, I would say, okay, these look like these are final stages, potentially final stages or beginnings of other stages. And so, the final result of mitosis, this is a conservative process. We expect to see one copy of each chromosome within it. So, the best answer for this would be F. I'd also note that there's two different metaphase twos. Metaphase 2, we know that the homologous chromosomes have already been separated into different cells. And so now we're going to be preparing to separate out the sister chromatids from each other. So I'd be looking for cells where there's only one copy of A and one copy of B, and they look like they're about ready to further divide. And so, that's a good answer for D. So we only see one capital A, one capital B, and it looks like the sister chromatids are getting ready to divide. And then you can similarly match B to the other metaphase 2 as well. We see that there's two different anaphases. So anaphase is active, so this is when separation has occurred. So we expect to see separation between sister chromatids. And so for this one, I would, again, come on, pen. I would match it with E. And the reason I would match it with E is because of the chromosome content. We have two copies of A, we have two copies of B, both homologous chromosomes, and they're starting to separate from each other. Anaphase 2, this you would match up with C, because we only have one copy of each homologous chromosome, and now they're actively starting to separate from each other. And so the last one to match would be prophase 1. And so prophase 1, the tetrad we expect to form in prophase 1. And so this right here looks like a good tetrad. The homologous chromosomes are paired together. And then the final result of meiosis 2, G, would be this, because we have a haploid cell with one copy of each. So if you were confused about the quiz, hopefully that helps answer some of the questions about where you might have gone wrong, and kind of how hopefully that you can start to see how the activities are are there to help you be able to answer these questions.